Hello, everyone. It's me, Brahm here. Welcome back to another episode of the podcast. Today's guest is Lars Wilde. Lars is the chairman and CEO and co-founder of Pangea Bio. Lars firmly believes in the power of nature to heal diseases based on the coevolution of the plant, fungal, and animal kingdoms. He started Pangea with the goal of discovering and developing molecules that have evolved with the ability to cure diseases with high unmet need. Prior to Pangea Bio, Lars co-founded two companies that the audience is probably pretty familiar with, Compass Pathways and ATAI Life Sciences. Lars, thanks so much for coming to the podcast. It's great to be here with you.
Bram, good morning. Great to see you, and thanks for having me.
Absolutely. It's my pleasure. So, okay, Lars, you know, most people I think already kind of know your background because you've been on a lot of podcasts before and people are familiar with Compass and ATAI. So I'm not going to ask you about, your whole life story and everything, but you already, you started Compass, you started ATAI. You're already making the rest of us look bad by starting two companies. Now you start a third company, Pangea. You're making me feel very unaccomplished. So why start a third company? What was the vision for Pangea? And, what was the reason that you decided to start this company?
Yeah, great question. And probably there are a couple of answers to your question. Let me riff on this for a little bit. So I think, first off, when I came into biotech, I was a biotech specialist, right? The companies that I had built before were in healthcare and in tech. And so I needed people to help me out and, climb up the learning curve very quickly. As one of these people was Dr. Stephen Wright, who was the head of R&D at GW Pharma, another company that has developed nature-inspired drugs in the cannabinoid space. Two drugs came out of that program, Sativex and Epidiolex. Sativex is a drug for spasticity in MS, and Epidiolex is a drug for severe forms of childhood epilepsy, Dravet syndrome specifically. And he became a personal mentor. We've always been talking about this, that there's this very interesting opportunity in drug discovery to go back to, I would say, the old days of drug discovery, but applying all our modern day tools to it in order to find novel, promising chemistry that can treat some of these intractable diseases that we still don't have an answer for, and neurodegeneration and neuropsych and related spaces. And so that effectively led us eventually down the path of saying, look, somebody needs to build a company around this thesis to look at traditional medicines with the help of AI, machine learning, and then find the most promising compounds and go after these large disease burdens. And hence the idea for Pangea was born. And obviously we had a lot of exciting people in our network that would help us make this company a reality. As it goes, companies grow up, and, Compass, where I was operationally running the business together with my co-founders, we hired amazing team members to come join us. Eventually, Kabir Nath, one of the leaders in the psychiatry big pharma space, to really take psychedelics through phase 3 and onto the market. And so for me, it felt a lot like, the job was done here in a way, and then somebody else had to take it all the way across the finish line. And I think this company, Pangea Bio, had to be built. Somebody needs to do this and take this forward because the promise is massive. And so we said, look, let's build this company. And here we are, two and a half years in.
Makes sense. So just to step back a sec, the high level goal of Pangea is to, you want to maybe go ahead and describe it for the listeners? Obviously, there's a lot to do with finding nature inspired medicines, or finding natural medicines and then figuring out how to optimize them for human and therapeutic use, which you kind of hinted at it earlier. You said, go back to the old days of drug discovery. Why is this something that has maybe been abandoned in more recent times?
Yeah, great question. And not obvious, to be honest. I think one of the things that I stumbled over that really was eye-opening to me is that actually 84% of all approved central nervous system drugs are nature-derived. And that includes neurodegeneration, neuropsych, pain. And that's obviously a massive number. That's by far the largest proportion in all disease areas. That's why we also said, okay, this is an area where we want to go into, because when you think about it, humans around the globe, Homo sapiens, depending on which anthropologist you trust, have been in existence for 300,000 to 400,000 years. So we had a lot of time to discover through trial and error plants and fungi and yeast that had profound effects on symptoms humans were experiencing. And if they indeed were efficacious and safe, people made them part of their medicinal arsenal, and initially passed them on through their healers, which would teach the next generation. And then over the last 200 years, a lot of that has found its way into literature, or in the 1,000 years before, sometimes into scripture. And so there's this treasure trove of information on these plants. Now, and that is how we developed a lot of the medicines we're using today.
Right.
Now,
the classic example is aspirin, right, which came from the willow bark tree back in, I think the early 1900s.
Aspirin is a great example. Yeah. And then from aspirin, people often don't recognize, but all the COX-2 inhibitor drugs in the pain space are derivatives of this drug, right? When we think about paracetamol, ibuprofen, etc., derivatives that stem from aspirin. And then when you think about pain medications at large, the opioids, right? The prototypical opioid is morphine,
of course,
from which then other opiates have been derived, and with a lot of problems down the road, but still one of the most widely used pain medications for very severe pain. I think there were a couple of technological advancements that received a lot of hype in the big pharma companies and big biotech companies. One was high throughput screening, the ability to use robotics and just screen millions of compounds in a short period of time in standardized assays against a certain disease target. So the focus went from, let's look at phenotypical expression of a disease and symptoms, to here's a target that's been validated. Let's hit that target as selectively as we can with chemistry. And so the idea was we can be much more specific, and we don't have to leave it to serendipity to find that one molecule that can treat a disease, but we can look at billions potentially of molecules, or millions is probably more accurate, in a short period of time to find a promising chemical. So there was one innovation where people said, look, why don't we don't have to send our scouts into the forests of this world to find plants and then spend years to figure out what is the one active in that plant. We can just take our libraries and screen them against targets. And then the other advancement was the development of in silico modeling. So the ability to model protein targets in computers. And that really started a whole field of people looking at computer models to predict the binding of molecules in order to change receptor constellations, be it as agonist or antagonist, to find molecules that would activate a certain receptor and to be very selective. Now, obviously, biology is very complex. And so what people had not considered back then is that it's very difficult to model any off-target effects from that, what the molecule might do. So in the early days, the hits were actually good, but then moving through lead phases and then into human trials, what we actually saw is that the chances of getting a molecule all the way through clinical trials into humans dropped significantly over the course of the last 40 years. So that is interesting. So while we had these developments in the 1980s, this didn't make us better at getting drugs across the line to treat people. Now, as I said, it's very difficult to do as part of the analysis that's required to actually figure out what's in a plant, right? A lot of PhD theses have been written about finding out one single molecule. Now, when we think about maybe a plant in our space that I worked in prior and that you're investing in, the psychedelic space, so Salvin RNA, the active in Salvia divinorum, a psychedelic plant in Middle America and Mexico, which had actually been analyzed at Sandoz Pharmaceuticals by Albert Hoffman. It took him years, and he didn't find out what the active was in the plant. And he's arguably one of the greatest chemists of all time, right? And it just shows you how complex that process was and why pharma eventually said, we don't have to go through this. We can be better than that. Now, the promise didn't hold up, and I think now we're actually at this point in time where technologies allow us to go back and look at plants at scale and still have an efficacy that is similar maybe to high throughput screening, but with a very different approach.
That makes sense. So the whole chain is sort of, you scour the literature, whether that's, ancient scriptures, ancient traditional Chinese medicine books, etc. You catalog all of these different ideas, and then you find the plants that those ideas are talking about, and then using some sort of new technology, you can figure out what actual alkaloids these plants and substances are made up of, and then you can get to work on optimizing those for therapeutic use. um, One of the big differences between a lot of these natural remedies and a lot of the like in silico design drugs that we've seen over the last few decades is that it seems like pharma is generally trying to optimize for like highly selective drugs, meaning they hit maybe one, maybe two, if you're lucky, receptors or targets very, very strongly, and they try to minimize any sort of off-target effects. But in the natural world, it's pretty rare to find compounds that hit one thing and one thing only. Do you think that that is maybe a reason why we've had a decrease in the success of clinical trials, because treating diseases is maybe not so simple as just targeting one thing?
Yeah, it's a great question, right? For some diseases, you want to be very selective, right? If you established a very specific target that has maybe a single gene location where you can hit a target, and you need to knock out a single protein. If that protein is knocked out, you solve the driver of the disease. That makes a lot of sense, right? And it's unlikely that if we're talking about such a unique case, that you will find the perfect molecule in nature. So I'm not saying one approach is better than the other approach, but if we think about the complexity of diseases in the neurospace, they are so multifactorial, right? We barely understand these diseases. When you think about some of the diseases that we're going after right now, Alzheimer's disease, Parkinson's disease, ALS, schizophrenia, these diseases are crudely understood. And then to my surprise over the last 5 years is depending on who you talk to. And these are well-published leading key opinion leaders. You might get very different opinions of what causes Alzheimer's disease or what is the major contributor to Alzheimer's disease. And I guess, my simplistic key takeaway is that it's not one thing. Probably all of these folks are partly right. And, so maybe something kicks off the disease initially, the pathology, but then you have contributing factors. Now, wouldn't it be great to have a molecule that actually addresses several of these disease drivers at the same time? And I think that is where coevolution and these naturally occurring molecules come in. And again, I would say two things on that to make it maybe a little bit more understandable for the listener. If you think about Epidiolex, the blockbuster drug that GW had created and Jazz Pharmaceuticals had bought for childhood epilepsy, the active is CBD, cannabidiol. And GW has worked very hard on figuring out what it actually does. And they made a proposal to the FDA, and the FDA effectively said, I'm butchering it now, but they said, look, you have no idea what CBD does. But when we look at the disease, it clearly works. So we don't care. So it might not even be necessary to understand what it does. Or on the other hand, you have something like psilocybin psilocin to treat mental health suffering, and you see that it's a very promiscuous drug that hits a couple of receptors reasonably hard when you think about serotonin 1 and 2 receptors, but is active across the whole range of the serotonin receptors, hits the sigma receptor, and now there's evidence that it also hits some kinases, which might have contributing effects to it. And so, when we think about SSRIs, where we try to be very selective, or several of the other approved first and second line treatments in mental health, what you find is that in these more complex psychedelic compounds, you effectively hit many of these targets at the same time, which probably contribute to their improved efficacy over current treatments. So, and then sometimes what we find is that there is a single target that we like to overemphasize. So I think that's also important to say. So we're not taking, botanical drugs forward per se, or we're not taking a natural compound forward. That's the starting point for our drug discovery work. So we find a molecule, we try to establish the mechanism of action, and then we try to dial up certain pharmacodynamic properties against disease targets, and that could be single targets or dual targets. But indeed, also our focus would be to be selective on something that we find drives the disease, but with the benefit of the efficacy data in human populations that gives us an unfair read, because we already have that evidence from human populations, and we don't have to figure out, what are all the off-target effects that might contribute to the benefits of the medication.
Right. Yeah, that is the benefit of starting with these natural products, is that in most cases there is this prior evidence in humans, which can really help focus your search and tell you which direction to go in. And your comment about GW Pharma not necessarily understanding the mechanism by which CBD does what it does is pretty interesting, and something that I think a lot of people don't quite understand is that the FDA doesn't care so much about why things work as long as they do work and they're safe. That's kind of what they're optimizing for. So let me ask you this. Let's just start at the beginning of the chain. You search through the literature, the natural history. What sources are interesting to use? Are you guys just scraping Wikipedia? Do you have some ancient text that you guys found in a cave somewhere that has a master plant list? What are some interesting sources that people could maybe look up on their own if they wanted to, just have a little rabbit hole to go down? Are there any cool books that you recommend?
Yeah, I think a couple of data sources are integrated into our databases. So they are definitely open source and subscription databases on the chemistry side of things, where we take known molecules and feed them into our databases with known PK/PD profiles and disease links. We feed disease databases into our databases. We use large language models, actually, that we trained to read and structure texts for us. So when you think about a lot of the texts that have been written about traditional use of plants are not in a table form. So it's very difficult to feed them into the knowledge graph. And I think that is one of these elements, one of the typical Bay Area VC question, why now? One of the reasons is that we can actually now handle these massive volumes of input data that we can then take, read, and structure. And we don't have to leave that to humans. We need to train the algorithms to do the work for us. That can also then be applied to scientific literature that we're feeding in. So it can go from someone writing a field report and just in the passing mentioning a couple of plants to a structured encyclopedia on traditionally used remedies, all the way to a scientific paper on a specific molecule that has been part of a traditional remedy, but is very much focused on receptor or transporter targets, etc. So all of that is then fed into our databases. And I think the last data source is actually our own work. So looking at LC-MS mass spectra output, to which we then apply machine learning. And based on the plant's unique spectra, or more specifically, the breakdown of molecules into certain spectra, we can use machine learning to uncover known and unknown chemistry in these plants. That might not have been published before. So that allows us to have a very unique set of data that we're then feeding into the knowledge graph. And I think the knowledge graph becomes the centerpiece where we can actually then uncover non-obvious connections. So where we can start, for example, with a disease, high-value protein targets in a disease, and then likely chemistry to hit these protein targets. And then we can look at where do these chemicals come from, where do they exist in nature, what was the traditional use of these plants. That is something that improves over time, because different tribes and peoples might have called symptoms very differently. So we need to aggregate them and understand them. But that allows us then to prioritize and go from millions of compounds that are in our database to a select few that we can then prioritize based on indeed strength of potency against a target, likelihood of or evidence of efficacy of the traditional remedy in human populations, and then looking specifically also at the molecules and asking ourselves, would that molecule have the right PK and ADME properties to be actually able to work with that molecule or even starting at can we synthesize this molecule, which at the moment is key to us. So we're not looking to use growing operations or any other advanced let's say algae genetic modifications to make a molecule, because that introduces another variable of complexity that we don't want at this point, right? We want to focus on chemistry and make the molecule. And so that allows us then to go down, hopefully, to a set of a handful of molecules, and that we can then take into the wet lab and test against the disease target, and from there into, for example, brain organoids or into animal models to show that indeed what we forecasted, what we know from traditional literature, actually translates also in the disease models in the animals, which gives us then further confidence that we actually found the active constituent in the plant that drives the efficacy of the molecule.
That's a great explanation. And the part that is, one of the parts that's most interesting to me is the bit where basically you've built this model that can look at the mass spec data and then predict what the actual compounds in the plant are. That seems somewhat novel, as far as I'm aware.
Yeah, it's a very novel field. That's the other bit of why now. I think the predictive value of the models being used has improved significantly over the past years, and there are not a lot of people in the world that are really good at what's called metabolomics analysis to find these plant metabolites. And so we have a phenomenal team within Pangea that is building our AI engine, and that are, I would say, world leading at concretely that part of the value chain and the discovery process. Which is great. And, sometimes that means, we would run our own mass spectra analysis. We would do it through partners, or sometimes we can also partner with people that already have the mass spectra from prior work in other fields. So we can just apply our technology to their mass spectra.
Yeah, so you don't have to be like Albert Hoffman spending years trying to figure out what's in salvin RNA. You can just run the model on the mass spec data, and then hopefully you will realize that it is the actual metabolites that are in there. That's very interesting. So, okay, so that's the whole value chain, or the process of the company. Let's get back to the indications that you're working on. Why choose neurological disorders as the place to start? Why is that the best place to start?
Again, a couple of answers. I think from the drug development perspective, I think we have the team and the competencies to actually develop drugs in the CNS space. And we've done that now multiple times across the team, including drug approvals and late stage drug trials. So we said, look, that is some key capabilities. So when we said, look, where do we want to start? It was pretty clear we wanted to start in the CNS space. Now, when you look at it more from, I would say, the scientific and likelihood of success perspective, then I would say it was very easy for traditionally living tribes to uncover plants, mushrooms, other naturally occurring molecules that would impact some type of their conscious experience. So if they were experiencing pain, it was very easy for them to see that they ate something or drank something that lowered their pain sensation, or if they were experiencing seizures or tremors, they would see that seizure frequency maybe dropped or the tremors went away. So that made it much easier for them to correlate the ingestion of something to the actual symptom. When you think about other diseases, such as, let's say, oncology, cancer, it would be much more difficult to say, look, I took a plant and I sense now that I don't even know what a tumor is, because I live in a tribe somewhere in the jungles of this world, and I realize it reduces my tumor size or slows down disease progression. Much more difficult to make that correlation, right? So we felt that that was a great starting point. And then, as you were saying, I think the complexity of the diseases in the CNS space is intriguing, because indeed we think a complex problem needs a complex solution. And I think that is something that nature offers. When you think about it, we're not only talking about the connection between Homo sapiens and these plants or mushrooms, but we're going back billions of years of evolution on this planet that was running in parallel, where effectively plants were optimizing or being optimized through the driving force of evolution against animals ingesting these plants for various reasons. And so you see that these plants either have constituents that or go back all the way to our very simple biology and connections between plant biology and human biology, or you see that these plants developed certain chemical mechanisms for two reasons: either to attract animals to go and eat these plants. So when you think about plant molecules that give taste and color, they oftentimes also have health benefits when we think about polyphenols, flavonoids, etc. Or you have molecules that want to distract animals from eating plants and mushrooms. And psilocybin is a great example. Psilocybin is in all likelihood a detractor, in that when you look at our serotonin system, the serotonin 2A receptor, which is responsible for the psychedelic effects, is nearly identical in flies and insects at large as it is in our human brain. So a fly that chews on a psilocybin mushroom in a forest will have some type of psychedelic experience that will deter that fly from keeping eating that mushroom. So you have that very interesting fit with biology through coevolution. And that is really what we're focused on, because we believe that has brought up molecules that indeed hit several interesting mechanisms in these CNS disorders that we can exploit.
And the mechanism that your lead candidate is going after is TrkB. Is that correct?
That is correct.
What can you tell us about TrkB?
Yeah, TrkB, I became very fascinated with a couple of years ago, because one of the effects of psychedelics is this profound increase in neuroplasticity and opening up of critical periods of learning. And I had fantastic conversations at the time with one of Compass's key advisors, and also now one of our advisors at Pangea, Dolan, who ran seminal studies that were published in Nature and Science on the ability of psychedelics to induce plasticity, metaplasticity in that sense, to open up these critical periods of learning that allow us to relearn habitual reactions to stressors and life events, allow us to change our response to a new environment. And that probably is a big driver of the effects that we're seeing with MDMA now nearing approval, and psilocybin, that people go through the psychedelic experience that gives them insight into their suffering, but then they have that plasticity to actually act on these insights and make changes in their lives. Now, that was fascinating, and I became fascinated with the idea of plasticity, because our brain is always plastic, right? We always react to our outer circumstances, and one can argue that these neurodegenerative diseases are inverse plasticity. And so we were wondering, what could we do? Can we actually drive plasticity in neurodegeneration to regrow neurons and synapses in order to reverse the damage that is being brought about by these diseases? And so that for us converged on a certain receptor, the TrkB receptor, which is the target receptor for the endogenous ligand brain-derived neurotrophic factor, which is a large protein. You can think of it a little bit like a growth hormone for the brain. So BDNF regulates plasticity, so the growth of new synapses, but it also drives cell survival, and it seems to have a potent effect as well on the clearance of, for example, misfolded proteins in the brain. Seems to also have an effect on mitochondrial health within the cells, which is another contributing factor in many of these diseases that we just spoke about. And interestingly, what is the smallest common denominator across the diseases that we are going after is a potent reduction in BDNF levels. That is something that also seems to happen in treatment-resistant cases of neuropsychiatric diseases. So when we think about treatment-resistant depression or treatment-resistant PTSD, we also see lowering of BDNF levels. I'm not sure how consistent that effect is, but we know that effect is very consistent in neurodegeneration. We also see this in schizophrenia. And so our question was, is there a molecule that actually might take over or might have some of the effects that we're seeing with BDNF? And I say some of the effects because We wouldn't want to have all the effects because BDNF doesn't only hit one receptor, but different versions, if you will, of BDNF hit different receptors, and some might be pro-apoptotic, something that you definitely don't want in neurodegeneration. And so the molecule that we found and decided to develop is indeed a small molecule modulator of the TrkB receptor, driving neuroplasticity. And so that is something that we've been focused on for the last years now in order to figure out what are the diseases that would benefit the most from modulating that receptor and driving up neuroplasticity.
That makes sense. Are there any like approved drugs or common supplements that modulate TrkB that people use commonly, or is this a relatively new target?
Yeah, there are none actually. And now you might ask, okay, what's the link then to traditional medicine? And in that specific case, we actually had it backwards, if you will. So here we started not with a plant in a disease symptom cluster, but we actually started with a receptor and said, this is a receptor we want to modulate. And from there, we were looking at, what are likely starting points for receptor modulation, and these molecules, where do they exist in nature around this planet, and have these plants that contain that natural molecule that we found been traditionally used in anything that is related to neurodegeneration and neuropsychiatry. And indeed, we found convergence in different populations that had discovered plants that included that specific molecule. That was our starting point. And they have been using that for a variety of diseases that are all connected to the brain. And so that gave us a lot of confidence, because when you find traditional use in different plants, in very different pockets on this planet, that evidence becomes much stronger, that you're not only dealing with a placebo effect. Now, when we talk about BDNF, so BDNF had been a hot topic in the late 90s, and several of our key advisors have actually been working at the time in Big Pharma with BDNF, because the idea was, we have this magic molecule, and if in a petri dish it treats all these diseases that we are going after, Alzheimer's, Parkinson's, etc. How about we administer that very molecule to the brain? Now, the problem with that is that it's a massive protein. It doesn't cross the blood-brain barrier. So you need to find a way to open up the blood-brain barrier to get it there. And then it's also a very sticky molecule, so it doesn't distribute easily in the brain. So even if you were to get it through into the brain, how would it distribute from there? So a lot of approaches failed. For example, Pfizer had a long-term partnership where they were using pumps, so they would drill holes into the brain, would actually put pipes into people's brain, and would actually administer a close cousin to BDNF, GDNF in that instance, which has a potent effect on dopaminergic neurons into the areas that are most impacted by Parkinson's disease. And in certain instances, in single patients, they found an improvement of the effects, but it was such a crude method that the signal went away in larger patient studies. So the protein approach here didn't work, and then also the antibody approach failed in various instances. When you think about the class of neurotrophins, there is actually an approved drug for eye disease based on GDNF that actually works. So when you think about the eye, it's the external part of the brain. So GDNF-derived drugs exist, but I think in that regard, we're pretty unique. Now, there's been a lot of excitement recently with the target. So it's one of these, I would say, undruggable targets, which is it's a word that
you're talking about TrkB here, right? Not BDNF.
Yeah, I'm talking about TrkB concretely, because and people declared it's undruggable. It's very difficult, to find something that actually modulates the receptor. Now, I think we're learning that, for example, these rapid-acting antidepressants, be it in the NMDA antagonist class, so ketamine and nitrous oxide, or psilocybin, mescaline, LSD, they seem to modulate TrkB. Now, there's a hot debate. There was a paper recently in Nature that actually seems to show that psilocin and LSD directly bind TrkB, and that even if you block serotonin 2A, you still get neuroplastic effects from these molecules. Now, in the space, there are also a lot of people that say they would want to see these studies replicated to really believe they are true, and maybe it's actually a different effect, that actually LSD does something else which downstream activates the receptor, which seems to be the case for ketamine, where you see that increase in plasticity for a short period of time. And I think what's understood with psilocin, for example, is that we actually see you administer psilocin and you see an increase in BDNF. But there is also the problem that, why are we not focusing on psychedelic scaffolds? Because it's very difficult, because indeed these are strongly polypharmacological drugs, and then indeed they have that behavioral effect. And when we think about these neurodegenerative disorders, we're talking about a chronic treatment. And so psychedelics would not work in that instance, but we have an interesting now class of molecules and families, first and second generation molecules that drive that plasticity actively. And so, we are very excited now to take this forward into the clinic. And we believe that similarly to, I would say, psychedelics, we have something that will not only work in a single disease, but is a whole platform across neurodegeneration and certain neuropsych conditions that we want to go after.
Right. You're still trying to figure out what the primary indication is going to be. Is that correct?
Yeah, and we're probably not going to narrow it to a single one. So in all likelihood, we're going to take this into several patient studies at the same time. And then the question is more, which ones are the right ones for the current molecule or the current pharmacokinetics and pharmacodynamics. So right now we've validated the efficacy in gold standard models of schizophrenia, Parkinson's disease, Alzheimer's disease, and in various in vivo models that allow us to also think about other diseases such as ALS. Now the question is, which ones of those do we want to, or subtypes of these diseases? Maybe we don't even want to go after Alzheimer's at large. Maybe we want to go after a subtype, or cognition and Alzheimer's disease, right? And so the goal is really now to drive the molecule class through the IND enabling tox work, which is gonna complete by mid next year, and then kick off the phase 1 studies. And I think the phase 1 studies are really exciting, because there we can do a lot of biomarker work, which would then allow us to really figure out, based on the biomarker signatures we're seeing, what are the disease areas that look most promising for molecule number 1, and which indications might we want to leave for certain biological reasons for molecule number 2 and 3 that are coming down the pipeline.
That makes sense. And this is maybe a good jumping point for my next question. So one of the core ideas here is that humans have sort of co-evolved with these plants, and that's why we're able to sort of work in symbiosis with these plants to treat diseases. Is it fair to speculate that maybe certain populations of humans have evolved with these plants more closely than others, and therefore there might be certain population subtypes that will respond better to a certain medicine, and therefore there might be ways to sort of segregate the patient population into different subgroups, that this group should maybe try this plant-inspired medicine, whereas this group maybe should not, just because of some sort of genetic expression. We already know that there's certain genes that give people a higher propensity for diseases like APO4 with Alzheimer's. Do you see sort of like genetic-based precision medicine as being part of the clinical trial selection? I know this is like way, way down the road and probably not something on the roadmap yet, but it's something that I'm curious about.
Yeah, I know, it's a great question. When we think about ApoE4 in Alzheimer's disease, for example, and double positive candidates, to see would we see a differentiated effect from the average Alzheimer's patient, right? And because that drives early onset of Alzheimer's disease, can we use this modality maybe even as a preventative treatment in high risk populations? All of these are amazing questions to ask. I think when we think about Alzheimer's disease, I would say in today's climate, probably we want to take this through the first patient study and then find the right partner to develop the molecule in Alzheimer's disease. I think that's where we're seeing a lot of interest already today. differentiated mechanisms in Alzheimer's disease. I think the field was very unattractive 6 years ago, when I was coming into neuro, and I realized that whenever I heard people pitch their story on their new asset in Alzheimer's, that VCs just shook their heads and turned around. And the field has come a long way since then, with the recent antibody approvals that are going after amyloid beta, showing slowdown of disease progression by 25-30%. That's a massive result. And I think that really revived the space and showed that indeed you can go and develop drugs that have a real benefit in patients. And so that's where you saw a massive influx as well back into neurodegeneration. And I think that's very exciting. So I think there's a lot of opportunity for partnerships down the road, because dementia trials are very long, very costly. And so I think that is something that in all likelihood we would want to do with a big pharma partner. And then there are indications that are much easier to run ourselves. When we speak about schizophrenia, ALS, Parkinson's, in comparison to Alzheimer's, much easier to run, still very difficult to run, but probably that is something that we would want to develop ourselves through all the way through to late stage clinical trials then.
Gotcha. Well, let me ask about sort of the final pillar of the Pangea ecosystem. When you're talking about using molecules in plants that have been known to certain populations for a long time, the question of impact comes up a lot. I know that's something that's important to Pangea. You guys are big supporters and try to always be in compliance with things like the Nagoya Protocol. Can you speak a little bit to the impact work? And maybe a good place to start might be, what is the Nagoya Protocol? I know that's something that probably a lot of people haven't heard of before, but it is something that comes up whenever you're talking about taking natural assets from different parts of the world.
Yeah, it's a very important protocol that was brought forward by the United Nations, effectively as a response to big pharma having massive benefits from taking plants, figuring out what the active molecules are, and making great drugs from it. But then the tribes that actually held the knowledge of these plants never benefited in the upside of the drugs that have been developed. And these drugs, had first of all a massive profit impact on big pharma, but then also massive societal value when they became off-patent drugs that are now widely used, right? When we think about some of the chemotherapeutic agents that are very widely used coming out of the jungles of this world. And so eventually people said, look, this is unfair. We need to solve this. We need to regulate this. And the United Nations established something called the Nagoya Protocol, which indeed regulates that genetic resources, if they can be traced back to a specific tribe or country, need to share in the upside of the development of the drug through benefit sharing agreements. And that is a very interesting mechanism to actually have a massive impact on these societies when done right, meaning that you want to work with the tribes, and if you develop a drug successfully, there would be royalty streams back into these communities that then can be used for infrastructure and educational investments in the region. And that is something that nearly the whole world subscribed to, minus the United States. But I think increasingly we're seeing that also US companies that are working with naturally derived molecules are voluntarily adhering to the Nagoya Protocol because it's the right and ethical thing to do. And that is something that we really like, because we believe that it's a unique opportunity that we have. And when we think about the different motives of why we started the company, a big one was also that we realized we're really dealing with the last generation of traditional healers and shamans around the world that have this vast body of knowledge in their heads, but there's no second generation of shaman apprentices that will learn from them and take this forward into the next generation, because through the internet, mobile phones, etc., the whole world is westernized, and people don't want to be natural healers anymore. Now, that also means that with them dying, the whole knowledge would be lost. And so I think the opportunity that we have is through our work to also play a small part in preserving that knowledge and making it accessible to the next generations. Because otherwise, it will be very difficult to discover the effects of these amazing plants. If we were to go through, we're not going to go through another periods of trial and error to find out the effects of these plants, right? And so it would be a shame if that knowledge was lost.
Yeah, absolutely. That's a good point. It really is kind of the last generation, which is sad. This is sort of a tangent here, but in addition to all of these sort of natural remedies, there's also this world of supplements. And one of the things that I think is kind of frustrating is that there's, thousands and thousands of over-the-counter supplements that you can buy, and everyone kind of has their own opinion about what they do. But because these substances have been around for a long time and you can't get any sort of IP protection on them, big pharma will never really run clinical trials to actually prove if they work or not. Do you feel like this is similarly like an untapped body of knowledge or chemical library?
Yeah, it's a great question. Not necessarily an area for us to exploit, but definitely a very interesting one, right? If somebody asked me, what company, if I wasn't building Pangea, what company do you want to build? I think that's a treasure trove of data that we have there, right? And the same mechanism that we're using would apply, right? And one of the great company builders in the world is Flagship Pioneering. The Boston-based incubator that also incubated Moderna and several other big successes in bio. And they have actually started a company that is doing that. They're effectively using machine learning to figure out what is likely driving the proposed efficacy of supplements, and then figure out, can this be turned into a medication? And I think the evidence.
Do you know the name of that company?
I should know the name
we
of that
can
company.
We can look it up later. It's okay.
Yeah. And I think that's an interesting approach as well. And there's great evidence, right? When we think about the field that we're in, right, in the neurospace, creatine, right, is, I think, one of the top 3 selling supplements on this planet. Definitely in the fitness industry, and people take it because it improves their work output in intense exercise. Right? That's how it became popular, and so it has mildly anabolic effects. Now, what the world has learned through great academic studies is that creatine actually has profound effects on the brain. So there are actually really well done studies from world-leading universities in mental health and neurodegeneration that show that constant use of creatine improves outcomes in such conditions. So, indeed, it would be a great source to start thinking about developing medicines. But as you say, I think the problem there is IP ability, right? I think it's no secret that in pharma, the currency is IP, so the patent you have on a given molecule. And that is very difficult in the space of supplements, because you find that in the supplement world, people try to create derivatives. I mean, there are gazillion versions of creatine, for example. And so it is actually extremely difficult to find something that you can create a drug from. And so that's very different to what we are doing, right? Sometimes we're talking about plants that don't even have a name, and nobody knows the chemistry. And so that's a great starting point to discover something truly novel.
Absolutely. Yeah. Thanks for going with me on that little tangent there. Just stepping back and looking at, the macro picture, the world has changed a lot since you were working on Compass Pathways and ATAI. Now we're in a world where interest rates exist and money is not falling from the sky. What have you learned about business, about starting companies due to these macroeconomic changes? And do you have any advice for people that kind of want to do things along the lines of what you're doing?
Yeah, I think the last 2 years have been really rough, right? I think, and a lot of entrepreneurs that started their first company in the last 4 years might have a wrong perception of how difficult it is actually to raise money and build companies, because cash was abundant, and it was not atypical that people would receive a term sheet after a 5-minute pitch without people ever having seen any documents of the company or anything, and these days are clearly gone. Now, I think that is actually advantageous because, when you think about it, I think there were over 50 CAR T companies being started in the last 3 years or 4 years. And, that it's unlikely that we need that many biotech companies around a single technological advancement. Rather, I would argue that you want to have the best people on that technology drive it forward. And yes, you want to have some healthy competition, but I think some of that was just exaggeration because of the availability of cash, right? And I think we're showing now how cash effective you can be. We seed funded the company ourselves, which was very helpful that we could do that. And obviously, that's not the situation for everyone. But I feel like that would not be necessary anymore in this environment. So we're seeing actually now that investors are aggressively looking for new investments again. So they actually want to see the good companies. So while in the last 12 months, I would say that the larger VCs were very much focused on their own portfolios and figuring out the companies that we've already invested in, what cash do they require in the next 24 to 36 months until the IPO window opens up again in all likelihood. How do we need to finance them? What do our fund resources look like? And do we actually have capacity to invest in anything else? And I think, that exercise has been completed. So people know where they stand now. And so we see them actually look out for new promising technologies out there. I think what's really difficult is the Me Too's. They will have a hard time. And I think we're seeing that in the psychedelic space where you see a lot of people going out of business and you see a lot of aggregation now and M&A happening, which makes a lot of sense, right? I think also the psychedelic space probably needs, 3-4 companies vigorously competing against each other. I think that makes a lot of sense because that drives innovation. And I think that will also keep costs affordable for the systems on these medicines. So I'm all for competition, but we didn't need 200 psychedelic companies, right? I think that was a bit wild. And so, I would say that you need to have something that is promising, that is differentiated, and then you need to find the right investors for you that are excited with what you're building, and then be in an area that is actually attractive. So I think something I personally invest and have invested quite a bit. You see a lot of people that have great science, they have great teams, but then they are going after something that is actually not a problem, right? When companies that have great new treatments for osteoporosis, which is a disease that's well treated. So you're not going to be able to entice biotech VC funds and pharmas to eventually invest in your company. So I think that is some homework that I would suggest for new entrants that are coming maybe out of grad school, postdocs that have worked on a great technology to really figure out, are they solving a problem for patients? And is it so much better than the standard of care that other people will get us excited around it? If yes, then money is available again. And I think that's something new, right? I would say the money came back in the last 3 months. So we've seen that investors called us up and said, hey, sorry, we missed your last round. Can we still come in? Which was cool to see because it's been a much different environment. And as you were saying, right, I think, interest rates, depending on which macro analyst you trust, will remain elevated for the next 24 months. Inflation seems to get under control in the US, which is the leading indicator for biotech. I think that's something that people should keep in mind, right? The moment we see a reversal in interest rates in the US, that will be priced into the public biotech stocks. And so you will see a reversal in the market at that point in time. If inflation keeps dropping the way it does at the moment in the US, I think, we will reach that point probably somewhere in the first half of next year. And I think, people are getting ready for that. You see that now that people are investing actively again in Series A's and Series B's, which is great to see.
Yeah, it's good. It's good for everyone, including my portfolio. So excited to hear that, at least from your perspective, the money is starting to come back. One other question just about sort of the macro trends. There's over the last decade or so, there was this excitement around bio and drug development platforms, whereas prior to that, oftentimes companies had just a single asset or a single set of assets, and there wasn't much talk about some sort of enabling technology. And of course, you are to some extent a biotech platform, so you will obviously have maybe a biased perspective on this. But what have you seen in terms of appetite and shifts in perspectives around like single asset, pure drug discovery companies versus these biotech platforms? I've seen a number of blog posts by people saying, the biotech platform is dead. And then I see another blog post that says, actually, the biotech platform is not dead. It's still here. What have you seen in terms of investor appetite and interest around the platform versus non-platform perspective?
Yeah, it's a great question, and I think the platform model. There are two definitions of it. As you were saying, I think the more accepted US definition of the platform is the idea of you have a central technology.
Right. That's what I'm referring to there. Yes. There's the other definition, which is you're sort of a centralized company that spawns companies, sort of like a ATAI to some extent. Yeah.
Like ATAI, for example. Yeah. And then there are some companies that are both. That in the gene medicine space that are a centralized holding that holds the IP on a certain gene medicine technology, but then they build subsidiary companies around diseases that use that technology. And so there was a hot area 2-3 years ago. And then I think it was dead. We originally thought about, should we start the company with a similar model and should we build it in that manner? And we actually said we're not going to do that going forward. Because we actually, with a focus on CNS, you can build a world-class team that can drive these different assets forward. So, but I was proven wrong. There have been 2 mega financings in the last 8 weeks to these decentralized platform companies from some of the world-leading biotech investors. So I feel like, if you have the right set of investors already backing you and you've chosen to have that model, for some people this works. Now I think that decentralized model doesn't make a lot of sense for us, and it creates a lot of complexity and incentive problems. Whereas, in the structure that we have today, the whole team is incentivized. Everybody shares in the upside of building a single company, and I think that has a lot of value. Now, on the technology question, I think that there was just an overreaction that you always see, right? People said, technology, we don't like technology platforms anymore. We like single assets that all we invest in, right? And so the market has turned, and now you see a lot of investments into technological platforms again, right? So that makes a lot of sense to me. Now, I think what is different now is what you cannot do anymore is you can't say, look, I have a platform and it's a crazy technology, and we have 50 scientists that are still working on the technology. We have no idea what we're going to do with it, but let us just sit here for the next 5 years and work on the tech. And then eventually, this will churn out some type of drug into an in vivo study 5 years down the road. And so you saw a lot of that, and you saw some of these companies go public, in the onco space. And so that created a lot of problems because, how do you value such a company in a down market, right? And it created a lot of confusion. So now what I think, and we're seeing that with our company, is people do appreciate the technology because they see that we can create a flywheel to keep creating novel medicines. But they're also very much looking at the lead asset and saying, look, this is your proof point, right? Does this actually work? Can you find novel medicines, novel compounds, and will you derive efficacy from them, and can you drive them into the clinic? So you need both.
Right. You need the asset and you need the platform technology. You can't just have these grand visions of building a platform that someday will produce something of value, hopefully. Got it.
Yeah.
Amazing. Well, Lars, this has been great. I appreciate you taking the time to share your wisdom with us. Is there anything you want to leave the listeners with? Any closing thoughts?
Look, I think, no grand wisdom will be shared by me right now. I think the what I really want to say is that, it's a privilege to be working in this industry. And I think, through all the struggles of the last 3 years or 2.5 years in a biotech crisis, right? I think the North Star that all these companies have is we want to make medicines for patients. And that's something great. Well, I feel like something like that can keep a team going and motivated even through the hard times, which I find great because that is not the same when you are in consumer industries or in tech. If you're working on something in a downturn that doesn't really work out and you have a hard time, if you don't have that North Star, it's much harder. And so I think that is something that unites the industry. And I think, we've seen a lot of a collaboration across companies where people helped each other. And, if companies overhired and had great talent, they shared that with the next company to make sure these people got hired really quickly into something else. When they were over-resourced. And I really like that in the industry. We're talking about biotech, and everybody thinks this massive industry, but then in the end, a lot of people know a lot of people, and so it's this great web of people that try to make something happen for patients, and that's great fun. And we're very grateful for the investors that back us. Obviously, Brom, you're an early believer in the field overall, and so
yeah, absolutely. That North Star is why we're excited to be investing in this space, why we're excited to be investors in Pangea. It's an exciting place to have fun and make an impact.
Absolutely.
Awesome. Well, thanks so much, Lars. Maybe in a year or two, we'll do this again and we can, get some updates on Pangea. But for now, I guess we'll say bye.
That'd be great, Brom. Thanks so much.
Bye.
Bye now.